Hello, everyone. Welcome back to Meds.Concepts. Today we are going to discuss an extremely important topic in molecular biology, replication in prokaryotes. DNA replication in bacteria is one of the best studied biological processes. Most of our understanding of replication mechanisms comes from E. coli. Replication ensures faithful transmission of genetic information from one bacterial generation to the next. In prokaryotes, replication is semi-conservative, bidirectional, highly coordinated, and extremely efficient. In this lecture, we will study oriC, initiation proteins, Replisome Organization, leading and lagging strand synthesis, DNA polymerase, Topoisomerases, termination, and replication timing in detail. Prokaryotic DNA replication mainly occurs in circular chromosomes such as those found in E. coli. Replication follows the semi-conservative model, meaning each daughter DNA molecule contains one parental strand and one newly synthesized strand. Replication is bidirectional. This means two replication forks move in opposite directions from a single origin of replication. Important components involved include oriC, DnaA, DnaB helicase, Primase, DNA Polymerase III, sliding clamp, clamp loader, and DNA gyrase. The entire E. coli chromosome of about 4.6 megabases replicates in approximately 40 minutes. Origin of replication, oriC. In E. coli, DNA replication begins at a specific site called oriC. The oriC region is approximately 245 base pairs long. It contains DNA binding sites and at-rich regions. The at-rich region is extremely important because adenine-thymine base pairs contain only 2 hydrogen bonds, making strand separation easier compared to GC pairs. DNA proteins bind to specific repeated sequences called DNA boxes. ATP-bound DNA oligomerases and bends DNA, leading to local unwinding of the at-rich region. Thus, oriC acts as the initiation center for bacterial replication. Replication initiation occurs through highly ordered sequence of events. First, ATP-bound DNA binds to DNA boxes in oriC. Second, this causes local melting of the at-rich DNA region. Third, DnaC helps load DnaB helicase onto single-stranded DNA. DnaB helicase then unwinds DNA bidirectionally using ATP hydrolysis. Next, Primase, also called DnaG, associates with helicase to form the primosome complex. Primase synthesize short RNA primers required for DNA polymerase activity. Finally, DNA Polymerase III Holoenzyme assembles, forming the replisome, replication fork and replisome. As replication proceeds, Y-shaped structure called the replication fork is formed. The replication fork is the active site where parental DNA strands separate and new DNA synthesis occurs. The complete replication machinery assembled at the fork is called the replisome. The replisome includes helicase, primase, DNA Polymerase III, sliding clamp, clamp loader, and single-strand binding proteins. Single-strand binding proteins stabilize unwound DNA and prevent reannealing. The replisome coordinates simultaneous synthesis of both leading and lagging strands. Leading and lagging strand synthesis. One of the most important concepts in replication is the formation of leading and lagging strands. DNA polymerase can synthesize DNA only in the 5' to 3' direction. because parental DNA strands are antiparallel. One new strand can be synthesized continuously, while the other must be synthesized discontinuously. The continuously synthesized strand is called the leading strand. It grows in the same direction as replication fork movement and requires only one primer. The lagging strand grows opposite to fork movement and is synthesized in short fragments called Okazaki fragments. Each Okazaki fragment requires separate RNA primer. In prokaryotes, Okazaki fragments are approximately 1000 to 2000 nucleotides long. DNA Polymerase III Holoenzyme. DNA Polymerase III is the major replicative polymerase in bacteria. It is a large multisubunit enzyme complex known as the DNA Polymerase III Holoenzyme. The alpha subunit performs DNA synthesis through 5' to 3' polymerase activity. The epsilon subunit performs proofreading using 3' to 5' exonuclease activity. The theta subunit stabilizes epsilon activity. The beta subunit forms the sliding clamp that increases processivity. The gamma complex acts as the clamp loader. DNA Polymerase III is highly processive, meaning it can synthesize long stretches of DNA without dissociating. Sliding clamp and clamp loader. The sliding clamp is essential for rapid DNA replication. In bacteria, the sliding clamp is called the beta clamp. It forms a ring-like structure around DNA and tethers DNA polymerase III to the template. Without the sliding clamp, polymerase would frequently dissociate from DNA. The clamp loader complex loads the beta clamp onto DNA in an ATP-dependent manner. ATP binding induces conformational changes that open the clamp and allow its loading onto DNA. After ATP hydrolysis, the clamp loader dissociates. This mechanism greatly increases replication efficiency and processivity. Primase and Okazaki fragment maturation. Primase is a specialized RNA polymerase that synthesizes short RNA primers. These primers provide the free 3' group required by DNA polymerase III. During lagging strand synthesis, multiple RNA primers are generated, resulting in multiple Okazaki fragments. These fragments must later be matured into a continuous DNA strand. DNA polymerase I removes RNA primers using its 5' to 3' exonuclease activity. It then fills the resulting gaps with DNA using its polymerase activity. Finally, DNA ligase seals the remaining nicks by forming phosphodiester bonds. Supercoiling during replication. As helicase unwinds DNA at the replication fork, torsional strain develops ahead of the fork. This generates positive supercoils or overwinding of DNA. If these supercoils are not removed, replication fork progression can stop completely. Therefore, cells require enzymes called Topoisomerases to relieve torsional stress. Supercoiling alters the linking number of DNA molecules. Changes in linking number are critical for DNA topology during replication. Thus, management of DNA supercoiling is essential for successful replication. DNA gyrase and topoisomerases. DNA gyrase is one of the most important bacterial topoisomerases. It is a type 2 topoisomerase that removes positive supercoils ahead of the replication fork. DNA gyrase contains Gyrase subunits, which cut and rejoin DNA, and Gyrase subunits, which hydrolyze ATP. Type I topoisomerases cut only one DNA strand and usually do not require ATP. Type II topoisomerases cut both DNA strands and require ATP. Topoisomerases poses a catalytic tyrosine residue that transiently forms a phosphotyrosine bond with DNA during strand cleavage. DNA gyrase is an important target of antibiotics such as quinolones and novobiocin. Termination of replication. In E. coli, replication terminates at a specialized termination region opposite oriC. This region contains several TER sequences approximately 23 base pairs long. These TER sites are recognized by the Tus protein. Tus binds TER sequences and blocks helicase progression in a directional manner. As a result, replication forks stop when they reach the Tus-ter complex. Eventually, both replication forks converge and replication terminates. Termination therefore occurs at specific loci rather than random positions. Decatenation and chromosome separation. After replication of circular chromosomes, the daughter DNA molecules often remain physically interlocked. This interlocking is called catenation. Catenated chromosomes cannot segregate properly during cell division. Therefore, bacteria use Topoisomerase IV to separate daughter chromosomes. Topoisomerase IV is a type II topoisomerase. It introduces transient double-strand breaks and passes one DNA duplex through another. This process is called decatenation. Decatenation is essential before bacterial cytokinesis. Replication timing in E. coli. One fascinating aspect of bacterial replication is replication timing. The complete E. coli chromosome requires approximately 40 minutes for replication. However, under nutrient-rich conditions, bacterial cells may divide every 20 minutes. How is this possible? The answer lies in overlapping rounds of replication. Second round of DNA replication begins before the first round is completed. This phenomenon is known as multifork replication. It allows extremely rapid bacterial growth and cell division. Finally, let us revise the most important high-yield points. Let us look at replication initiation first. Prokaryotic replication is semi-conservative and bidirectional. The oriC region is rich in at pairs. ATP-bound DNA initiates the replication process. DnaC helps load DnaB helicase onto DNA. Primase or DnaG synthesize short RNA primers. DnaPol III Holoenzyme acts as main polymerase. Shift focus to the middle column. The beta clamp increases enzyme processivity. Clamp loading process strictly requires ATP. Lagging strand is made as Okazaki fragments. DnaPol I removes primers and fills gaps. DNA ligase seals remaining nicks between fragments. Finally, let us discuss topology and termination. Helicase activity creates positive supercoils ahead. DNA gyrase or type II topoisomerase relieves it. Replication terminates at specific TER sites. Tus proteins bind firmly to these TER sites. Topoisomerase IV decatenates the mixed daughter chromosomes. E. coli utilizes overlapping replication cycles. These are highly important concepts frequently asked in competitive examinations. Thank you. If you found this video helpful, please like, subscribe and share this video to your friends.